Happy day to all. Today we are going to start chapter 8, class 10 biology. How do organisms reproduce? Day 2. So last class we have seen natural vegetative propagation. And today we will start with the artificial vegetative propagation. So where the picture itself it shows how the base stem cutting take place, and then how you can artificially propagate the plant. So when a plant part is cut from the parent and put into the soil, which later give rise to a new plant. And stem cutting, it can happen in rose, sugarcane, and root cutting, it can happen in dahlia. The next method is layering. So the three types of layering you can see: simple layering, mound layering, or otherwise called as stool layering, and then the air layering. Before moving on to that, I'll tell you what is a layering. So a new plants are formed when stem of a parent plant, without detaching them initially from the parent plant, will get form a new plant. So here the three different types we will see the simple layering. So here we can see the bark of a plant that emerges downward towards the soil part, get buried under the soil, and after a few days or a few weeks we can see small root arising from that part. And once the straightening up of that bark part take place, they will cut the part of the bark that moves out of the mother plant, and then two new plants will be produced. So the second type, that is the mound layering, where you can see a heap of soil will be dropped on the stem towards the stem part of the plant. When the stem part comes into contact with the soil, there they arises these root parts. So you can detach from the parent one, and then you will get new plants by mound or stool layering method. And the third one is air layering, and also called as a Chinese propagation. So area of a plant is girdled and then surrounded by a moist growing medium that is sealed with a polythene film. So it has been, the bark part has been removed, as well as providing all nourishments will be covered by a polythene cover. After few days you can see small roots arising from that, and this method we call it as air layering. And a variety of plant can be grown by these methods, that is in strawberry, guava, lemon, China rose, etc. The next technique is grafting. So where here in grafting, the stems cut from the two different plants, they are being attached to grow as a single plant. So which helps, what is the significance means it improves the quality of certain plants. So for example, we have two mango trees of same species, where one type they don't have uh, that is the strength of the stem is comparatively less. It cannot withstand the fruitlets coming out, and then it will get removed from the parent plant, usual situation. Whereas yield, the other one, where here in this the yield will be too high, whereas another mango tree, it has got a strong stem part or bark part. So in such cases we choose two parts of these two different plants. One, it is the stronger character that we need to choose. We should take the bark part, that part we call it as the stalk. And uh, that is where it has got the strong bark part. The next one is the high yielding mango part. From that plant also we use a part of it will be cut and then will be wound with twine, and then the connection between stalk and scion is clearly given in this picture, and then tied and left. So after few weeks, we can see that the thing that are wrapped and waxed above part start producing leaflets, and then the plant here which shows the stronger base part or the bark part, and then the character of the high yielding variety will be produced. Next technique is tissue culture, or otherwise we call it as the cloning tissues, where this is a new technology that came forth for the production of enormous number of plants from a group of tissues, we should say. So small section of these plant tissues, it can be cloned using the process called as a tissue culture, and it is carried out in an aseptic condition, that is in a sterile condition inside the laboratory, and you can go grow enormous number of plants from these tissues. So here the capacity of a plant to produce their own, that is the plants from their tissues, that characteristic feature we call it as totipotency. So here that totipotency is being involved in the tissue culture. So what takes place in this tissue culture or cloning of tissues? From a plant in which we need to produce thousands of similar plants from one cutting, um, what we have to do? We have to extract tissues from those plants, which will be chemically treated with the hormones inside the laboratory in a petri dish. These tissues will be placed clean, aseptic condition, um, providing all growth hormones will be provided, and normal temperature for the plants to divide or those cells to divide. So within 15 days, you can see a huge mass of millions of tissues formed. That is, the petri dish will be filled with cells or tissues. Again, they separate those tissues into different petri dishes, and they provide all the hormones uh, required for the growth of the plant. And after another 15 to 20 days time, we could see the huge mass of tissues formed, and when these tissues are provided with plant hormones like auxins, gibberellins and all, we can see that it develops into a new organism like this. At a stretch, thousands of that new plants can be produced, and they grow into a new plant, as well as uh, the characteristics are being guaranteed. Next, we should pass on to the sexual reproduction in organisms. So sexual reproduction in which the new individuals are formed from two parents. Here, I would like to stress sex is involved. That is, the male sex as well as the female sex, they produce their own gametes, and these fusion of this male gamete and then the female gamete give rise to a new organism. So the new individuals are not identical to the parents. Variation occurs because it is a sexual reproduction, and they are relatively slower mode of reproduction. And why this sexual reproduction? So already we have seen in plants, the plant body itself can produce. So they cannot withstand for a longer generations. They will die because the genetic materials are not being combined, or a newly formed, or variation will not exist, will not result, and no variation, then the deterioration of the species will take place. That is why we need sexual reproduction. So why? So to incorporate variation in the species, which in turn ensures survival of the species, and each individual in a species has its own uniqueness and identity. So here you can see eye color chart as well as the human beings. So human beings, as we know that they north, south, or whichever country or whichever continent, we can see with lots of difference in their physical stature, then too we call them, they are Homo sapiens. So next we should see what makes unique, or the same characteristic feature in the generation after generation, whether it is a plant or whether it is an animal. So we know that if we plant papaya seed, we get only papaya plant. If we sow seeds of uh, mango, we get only mango tree, or from that we get only mangoes. So why? So there are some factors that are responsible for copying characters from parents to their young ones, and this, the thing which copy the characteristic features of the parent to the next generation, we call it as DNA, that is deoxyribonucleic acid, which is present in the chromosome, and these chromosomes are responsible for carrying their own traits, different characters. What do you mean by traits? They are the characters that are present in that organism. So if a parent, father and mother, both, they have their own characteristic features, so those characteristic features need to be passed on to the next generation, and this passing up of these general characters, inbuilt characters, takes place by means of the DNA, or the genetic material present in the cell, that is nothing but the DNA. So here I have showed how an organism is formed. So inside the organism we could see the cell, and inside the cell we have the nucleus, and inside the nucleus we have a chromatin network, and during the process of cell division, these chromatin networks condenses to form the chromosomes, and these chromosomes are the storage system for uh, uh, transferring uh, genetic traits from generation after generation. And for the sexual reproduction, we require gametes. So I said female gamete as well as male gamete should be produced, or otherwise these gametes are called as sex cells. And in some organisms, the male and female gametes are identical to each other, while in some other, male and female gametes are different from each other. The next thing, it is the male gamete, they are motile. Female gamete, they store food. And when the fusion of the male and then the female gamete will give rise to a new organism. So in this pictures, you can see the different types of uh, anisogamy, that is anisogamy of motile gametes, and oogamy, that is egg cell, oogamy, and then the motile sperm cells. And anisogamy, they are non-motile gametes. So here you can see the chlamydomonas, a male and female gamete, where it is not that much we cannot identify. Whereas the bottom part, the yellow one, and then the sperm that we could see, it is the human ovum and then the human sperm. And this is when the sperm gets fertilized with the or fused with the egg, we call it as sexual reproduction. So next we will see how sexual reproduction takes place in plants. The main reproductive organ of a plant is the flower. A flower is said to be unisexual or bisexual. If either male or female reproductive part is present in a flower, we call such flowers as unisexual flowers. Example, watermelon, papaya, etc. Where a male plant as well as female plant is present, where the male plant produce only male flowers and female plant produce only female flowers. Whereas, so here you can see the picture of watermelon, male flower as well as the female flower. Male flower you can see at the base of the thalamus part, below the thalamus part, petiole part, you don't have a swelling part. In female flowers, you can see the watermelon like small swollen part of the watermelon female flower. The same way, here you can see the papaya flower, male flower as well as female flower. And this is the normal hibiscus flower, where you can see both male as well as female in the same plant. And if both these male and female reproductive part are present in the same plant, we call it as, or in the same flower, we call it as bisexual flowers. Example, China rose as well as mustard, etc. Next, it is we will wind up what are these asexual reproduction. So asexual reproduction, it is easier and faster mode of reproduction. Within weeks time, you can produce new plants. It is stretch, and it is useful for seedless plants, can be grown by this vegetative propagation. And also, if we need any desired traits to be preserved or brought into the next generation, we can go in for asexual reproduction. Then the new plants formed are genetically identical to the parents. And some drawbacks too are there, where they are easily prone to diseases, more prone to diseases, and no genetic variation, which in turn what will happen generation after generation, it fades off or it moves off. Whereas if it is a sexual reproduction, they are relatively slower mode of reproduction. Genetic variations are seen and they are less prone to diseases. When we are going on to the sexual reproduction, the first main thing we need to know the structure of a flower. The basal part, we have a swollen upper portion of the stalk. We call it as the thalamus part. Then we have colored part of a flower which attracts insects for pollination, etc. So we call them as petals or otherwise called as corolla. Next, it is the sepals. Sepals are the green colored part, otherwise called as calyx, ensures protection when the flower is in the budding stage. Next, it is the stamens, which forms the male reproductive organ or part, which consists of anther and filament. The male gametes, they form the yellowish colored pollen grains, which has got two parts, that is a bulb-like structure called the anther and a stalk-like structure called the filament. Next comes the carpels. So the carpels, they form the female reproductive organ. The female gamete forms the egg or the ovum, or otherwise we can call it as ova. So here you can see the structure of a stamen or a carpel. So the first diagram, it shows the structure of a pollen grain, which has got two parts, a green stalk. This pollen grain, it consists of a filament, the green colored part, and an yellow colored part, a bulb-like structure. We call it as the anther. And in between you can see a connective part, a connector. So this is the cross section of this pollen grain, where you can see pollen sacs, four or two pairs of pollen sacs are present. This is the structure of a carpel, and then the structure of a stamen. We have stigma at the top with a thread-like structure called the style, and at the basal part it is the swollen part, we call it as the ovary, and inside that we have the ovules. So this is the internal structure of the when we cut open, we can see the stigma on top, style, and then the ovary, which consists of the ovule inside, which constitute the egg cell, integuments, egg cell, and then the two polar nuclei, and then the nucellus. How does sexual reproduction occurs in plants? The stages or different series of the processes that takes place in sexual reproduction in plants. They are pollination. First, pollination should take place. Next step is the fertilization. After the fertilization, the seed formation, and then the seed germination. As we are going to learn about all these processes. The first one is pollination. What do you mean by pollination? The transfer of pollen grains from the anther to the stigma, we call it as pollination. And when we talk about pollination, it is of two types, self-pollination and cross-pollination. Self-pollination, when the pollination takes place in the same flower, that is in a bisexual flower, we call it as self-pollination. When the pollination takes place between two different flowers of the same plant, or of different plant of the same species, we call it as cross-pollination. Next term that we need to learn here is unisexual, where we know that male as well as a female part is present in different flower. Then the cross pollination, that is earlier we have seen watermelon as well as papaya. They have male plant as well as female plant. There we cannot have self pollination. There they need to stick on to this cross pollination only when the flowers are unisexual in nature. So this transfer of pollen grains occur with the help of um, some external factors or external pollinating agents like wind, water, insects, and birds. So when these birds or flies, they approach, seeing the bright color or scent, they could smell the scent, they move towards these flowers for taking nectar. And while taking nectar, the pollen will get stuck on their body parts, and when these flies move from or insects move from one flower to another flower, there is a chance of transferring this pollen grains that got stuck on that insect to the stigma of the other flower. When the fusion of these male gamete with the female gamete take place, we call it as fertilization. Fertilization is the fusion of pollen and then the egg, we call it as a fertilization. And the result of fertilization is the zygote formation. So this picture, you can see the structure of pollen grain fallen on the stigma. When it falls on the stigma, the chemicals present in the pollen grain will rise off, will melt the outer exine part of the pollen grain and then starts germinating through the style of the uh, flower or the stamen. So as a result, what takes place? These things, they move throughout the style and then enters through the micropylar end into the ovary or the ovule, where female germ cells are present, the fusion of the male gamete and then the female germ cells takes place, resulting in the zygote formation. And what takes place? So here in such plants, double fertilization takes place. That is um, when one male germ cell fuses with the egg cell, and another germ cell, when it fuses with the polar nuclei, it forms endosperm as well as zygote, which results in double fertilization. So once fertilization takes place, the zygote will be formed, and this zygote divides repeatedly to form embryo. And ovules develop as a thick coat and form seeds, which is these seeds are embedded inside the ovary, and ovary ripens to form the fruit. So here the seed formation takes place. A seed or the seed will contain a dormant plant embryo and will have a food reserve, a protective covering, and each fertilized ovule develops into a seed. And there are two different types of seeds, that is endospermous seeds as well as non-endospermous seeds. The food reserves, if it is outside the embryo as a triploid, we call it as endosperm or endospermous seeds. Example, rice, maize, etc. Whereas a non-endospermous seeds are those seeds, the food reserves is within the leaf of the plant embryo. And these beans, peas, etc. are examples for non-endospermous seeds. So this is the structure of the dicot seed as well as the monocot seed. And in the practicals, you will be learning about the structure of the dicot seed, which consists of two cotyledons, the seed coat, and then you can see the plumule as well as the radicle that emerges out. So the radicle develops into the root system, and then the plumule into the shoot system. So this is the structure of monocot seed, which consists of endosperm, where the plumule and then the radicle is seen, and the cotyledons are embedded inside. So here how the formation of seed has been explained. So after the fertilization taking place, what happens? The germ cells get fused with the egg cell as well as the cell forming endosperm and then the embryo, and this which will develop into the seed, that is the developing seed you could see here. So next it is how the germination takes place. So the development of a seedling from a seed, we call it as the germination. So here you could see the process of germination. So when the seed is dropped inside the soil, within 2-3 days time, you can see a small white hair-like structure emerging out. That is a breakage of a seed coat. It emerges out. We call it as the radicle, and this radicle it develops into the root system. Next, it is the hypocotyle which will move out, opening the seed coat will be split open, and then by that time, small the epicotyle will emerges out in between the two cotyledons, which results in the formation of a new plant that is emerging out of the seed. This is how the germination of seed take place. So how here the complete process is being shown, where how the germination of pollen grain it takes place. From there, the growth of the pollen tube through the style which enters, get fused to the sperm and then the egg forming embryo. After the fertilization, the zygote develops into which are having embryo, and then this embryo young plant will be present inside the fruit as seeds, and then when the seeds are being thrown out, or after consuming the fruit, we move the seed out, and when the seed falls on a suitable atmosphere or substratum, it develops into a mature sporophytic plant.